next speaker. It's Randal Koene, who just spoke. He's a neuroscientist at Bokta and the co-founder and chair at the Carboncopies Foundation. Dr. Koene is a globally recognized neuroscientist for his work in neural interfaces, neuroprostheses, and the functional reconstruction of neural tissue. Today, he'll present on what is called the inverse problem. Let's give a warm welcome to Dr. Koene.
Thank you. And I should correct, I'm not at Voxa anymore. So that's in the past. Right. So this is going to be a little bit different, mainly hoping to drag the conversation into, I guess, what's more of a speculative area. The points that I'm going to be going through on these slides are a summary excerpts of a draft paper on the inverse problem of brain emulation. And I'm trying to do the topic justice gradually, even though I freely admit that I'm not a world expert in mathematics of inverse problem solving, control theory, system identification, or complex nonlinear systems. So I wholeheartedly invite critical feedback, recommendations, or hey, if you're feeling truly inspired, co-authorship. In any case, let's ease our way in with some basics so that we can fall back on those fundamentals when we consider a specific problem. And of course, for some of you, this is all old hat. So, physical systems. We can express the study of physical systems in terms of three steps. Parameterization of the system, that's discovery of a minimal set of model parameters whose values completely characterize the system from a given point. Or forward modeling, discovery of the physical laws that allow us to, given values of the model parameters, make predictions of results on measurements for some observable parameters. Or inverse modeling, using the actual results of some measurements. So I was pointing out these three different steps or ways that you can study a physical system. There's some model space, which is a manifold of all the possible model parameterizations. And then experiments typically suggest physical theories, and physical theories in turn predict outcomes of experiments. And comparing a predicted outcome with an observed outcome enables amelioration of the theory. If we include in a physical theory the physical parameters that describe the studied system, then the inverse problem theory concerns the qualitative rules to be used for such a comparison between predictions and observations. System identification in brain emulation involves finding parameter values for the mathematical representations of brain processes within models that compose a runnable emulation. There should be a set of possible representations that together satisfy a particular set of success criteria. The representations in that set will have a multitude of parameters, and if well chosen, then the parameters may be quantified or tuned using data that's obtainable from a biological sample. You have to find a solvable relationship between these observed data and the parameters that need to be determined. When using structural, say morphological and connectome data, the system identification problem is addressed by deriving structure at an appropriate resolution and then solving the functionalization of corresponding models. In general, and in biological domains like brain emulation, inverse problems correspond to the particular case where the data space, D, and model space, M, have fundamentally different physical meaning, and where we're interested in translating information from the data space into the model space. In system identification, the inverse problem refers to the process of determining the underlying system parameters or model structure based on observed input and output data. You try to attempt to reverse engineer the system. You also refer to the system as the forward problem. In general, you can express that forward problem as shown here. Y is some f of x plus epsilon, where x represents unknown parameters like system properties, initial conditions, inputs, and y represents observed data, so measurements, outputs, effects, and epsilon represents noise or observation errors. And then f is a mapping from parameter space to observation space, and you infer the characteristics that produce the measured data rather than predicting future outputs from a known system model. The inverse problem involves solving for x, which is confounded by the following issues. It could be an ill-posed problem if multiple system models can produce similar output data, making it difficult to uniquely identify the correct system parameters. Or the inverse problem may be ill-conditioned due to noise sensitivity, meaning that the accuracy of the identified model is impacted by real-world data with significant variability or by corruption by noise. Solving an inverse problem can also be a means, for example, to optimize a control system. In essence, all or a very large proportion of scientific problems could be described as identification and control problems, and identification problems are problems of cause and effect. In fundamental neuroscience, much of the research involves problems of system identification, even though it's not always posed as such. Consider, for example, the relationship between two synaptically connected neurons and the question of how a presynaptic action potential causes postsynaptic potential, or at a more macroscopic level, how do the photons that are getting into my eye cause neuropopulation activity in the visual pathway? And this obviously goes beyond simply reporting experimental results. As first specified here by Jacques Hadamard, an inverse problem is well posed if, for the system, there's a solution, and that solution is unique. The solution is stable. In other words, it hardly changes when there are slight changes in initial conditions or parameters. The solution is not highly sensitive to small changes in Y. And then solving that inverse problem typically involves an analytical, statistical, or control theory approach. A review of inverse problem theory by Villaverde and Donga that was applied to reverse engineering and identification in systems biology highlights a few important lessons. One, modeling should begin by asking questions about the intended use, the purpose, goals, even if only approximate answers are possible. While deterministic methods shouldn't be abandoned, approximate methods can give rather good solutions to many hard problems at reasonable cost, and there's great promise that hybrids between deterministic and stochastic methods can work well. And we need to exploit the structure of dynamic models. In inverse problem theory, the smallest eigenvalues of a system matrix play a crucial role, as they often indicate the level of sensitivity to noise and instability in the solution. This means that a small eigenvalue can lead to significant amplification of errors in the reconstruction process, making the inverse problem ill-posed and difficult to solve accurately. Small eigenvalues represent directions in the solution space, where small changes in the data can lead to large changes in the reconstructed solution. The main problem in system identification is to find a suitable model structure for which a good model can be found. Parameter estimation is typically a smaller problem. A primary rule is not to estimate what you already know. Use prior knowledge and physical insights when selecting model structure. As an aside, it might be worth noting that there are quite a bit more in the sense of prior knowledge and available data that could be used than what has been used in, for example, studies just presented to produce results, and some of them are listed here. I won't go into details. 3 degrees of prior knowledge are typically distinguished. White box models, that's a model where, it's perfectly known it was constructed entirely from prior knowledge and physical insights. Gray box models, where some physical insight is available, but there are parameters that have to be determined from observed data. And then black box models, where no physical insights available. Model structure is chosen based on flexibility and performance. To understand where models used in brain emulation might fit in, it's useful to further subdivide the category of gray box models and note where black box modeling belongs within those. There's physical modeling. The model structure is based on physical grounds, leaving a specific number of parameters to be estimated from data. And there's semi-physical modeling. Physical insight suggests certain nonlinear combinations of signals for which measurable data is obtainable. Those signals are then subjected to model structures of black box character. Below, we'll see how system identification and brain emulation carried out by functionalizing models constrained by connectome and additional knowledge fit into that category. If system identification for brain emulation involves gray box modeling on the whole and semi-physical modeling with black box system identification at a component level, these are overall systems that contain many smaller black box models. We can then ask how small should those black box models be? And we'll see below that this question ultimately comes down to the problem of correct goal-directed scale separation. Before we dive into these conceptual issues of primary importance, let's formalize the black box problem a bit. For each nonlinear black box problem, we can write the observed inputs ut at each discrete time point, the observed outputs yt at each discrete time point of a dynamical system, and we're looking to solve yt as a function of g, where g gives us a good prediction of yt. and Vt should be a small additive term that adjusts for the fact that Yt will not be an exact function of past data. Here, theta is a vector of parameters, as we find function g within a family of parameterized functions. And the quality of theta can be addressed in terms of the fit between model output and observed data, where the particular norm and minimization are problem specific. It's useful to decompose G into two partial problems. So how to choose the nonlinear mapping from regressor space phi to output space, and how to choose the regression vector phi from past inputs and outputs, mapping UT and YT to a regression vector that is itself parameterized. The way in which regressors are combined in G will clearly reflect the structural assumptions about the system. Let's assume that we understand our forward problem well enough to choose an appropriate level of description, and that we already used physical knowledge to decompose the overall problem. For example, in one of the ways shown here, but not limited to these, as a system of point neurons, or cells described in compartments, or a combination of levels, perhaps with explicit molecular interactions. This is determined by our physical understanding of the system and can be by the feasibility of solving the remaining black boxes, being able to estimate regressors from recordable data. There are many ways to solve those estimation problems at the component level. We're not going to detail the many choices available here, such as wavelet and radio basis networks, kernel estimators, multi-layer networks, recurrent networks, and many more, and instead refer to the literature. But it's notable that a useful approach may be to build a linear model first, such that the residuals contain all the unmodeled nonlinear effects. You can then use a neural net, deep learning, or something like that to treat those residuals, and the linear model is robust, and the resulting nonlinear model is then at least as good as the linear one. It's preferable to use physical insights to build up nonlinear effects in a model because this can typically be done using fewer parameters. And ideally, model residuals should be white noise, but note that what we consider noise or irrelevant depends on the context of a model, in other words, the meaningful function. In order to even have a shot at specifying and then solving a well-posed inverse problem, one ought to clearly understand the problem that one's seeking to solve. What is the output or response Y that you need to correctly predict, model, replicate, emulate? What are the relevant input, stimuli, or source conditions X to do this for? And what do you need to care about in the context of a particular set of aims? Summarizing, to reach a successful solution, you need to define aims concretely, identify relevant signals, information variables that matter to those aims, properly state the corresponding problem. Then you can solve the inverse problem for a specific data set by studying mechanisms that operate on those signals, by representing, abstracting, and modeling them, and by ensuring that the problem isn't ill-posed or ill-conditioned, that it's robust and generalizes adequately. In well-developed scientific domains, specific goals are clearly stated and problems therefore concretely formulated. Theoretical work and reviews pertaining to brain emulation are typically not yet as rigorous, making do with more implicit goal statements. Every attempt to work towards a useful theory by specifying a representation for a hypothesis, a model, carries within it an explicit or implicit separation of scales that's intended to best suit a purpose. But why worry about specific goals, scale separation, and the associated data resolution at all? An alternative approach could be to set aside those concerns. After all, they may be difficult to resolve with our current scientific understanding of information processing in the brain, and to instead try to build an emulation all the way from the ground up. After all, a fundamental assumption behind the concept of brain emulation is the functionalist and mechanistic idea that every property, every behavior of brain and mind is an emergent result of the totality of component operations and interactions. So an emulation achieved by complete and correct model reconstructions of every component in the brain tissue, down to the molecular, atomic level if you want, ought in principle to satisfy every conceivable emulation goal. Emulation involves representing operations and operands from one system of physical components via operations and operands in some other system of physical components. It's a conversion from one system of representations to another. Both systems involved ultimately rely on the same physical elements and laws, of course, but the representational primitives of neurobiology and of electronic computation are expressed differently. Conversions like those have been studied extensively in mathematical theory, and in all but trivial cases, there's a cost associated, an unavoidable inefficiency that can be described and indicated by calculating the Kullback-Leibler divergence or by means of a Kantorovich-Wasserstein metric, among others. Already, today's relatively incomplete models of neurobiological circuits consume far more power to compute than their biological inspirations. It's a contrast between a human brain operating at 20 watts to 40 watts or something, and compute clusters demanding kilowatts or megawatts to achieve something approximating real-time performance for somewhat realistic models. It's immediately obvious that merely based on the requisite power budget, a total depth emulation at, say, something like human brain scale, let alone many human brains, that approach is a practical infeasibility. Setting that aside, as it may be inconsequential for a first brain emulation, there's a far greater practical problem that obtaining the necessary data for a correct total depth emulation is currently impossible. It's quite likely that it will remain infeasible or far too costly, even in the long term, for emulation of brains beyond those of very small organisms. Even if that challenge can be overcome, there's a more fundamental problem with reliance on total depth emulation. Doing so actively hinders or prohibits the intended outcomes of brain emulation for medical and neuroprosthetic goals. To mention just a few undesired consequences: reduced robustness and efficiency of the resulting emulation, susceptibility to models of brain disease, susceptibility to the deleterious consequences of aging, as those would also be in there. So I'm positing here that medical and neuroprosthetic goals of brain emulation are best served by an emulation that has the following properties. 1, the emulation efficiently retains necessary function, the emulation has a footprint that's as light as possible, and the emulation has abstracted as much of the original substrate as possible. Okay, let's begin to formalize the problem of brain emulation so that we can begin to clearly distinguish problem statements for brain emulation with specific goals and success criteria. So let the general form be what I'm showing here, y equals fx plus epsilon again, but this time of chi, where the subscript chi denotes a sample space relating directly to the particular focus level, that's this LG, of emulation at which the majority of success criteria, CG, for one's emulation goals, G, are to be verified. With this, we immediately begin to see that what will be the principal challenge discussed in the following slides, namely gaining a rigorous understanding of the problem in terms of its goal-dependent sample space, and consequently the observed data y chi and parameters x chi that are most relevant to that space. In the discussion of black box system identification, we already indicated that brain emulation may be best described as gray box semi-physical modeling, where a model is a composite arrived at by applying as much physical knowledge as can be obtained via high-resolution, high-throughput techniques, with a resulting set of many smaller black box models to solve for system components. And that's the so-called functionalization step. As always, one must strive to avoid ill-conditioned problem solutions caused by overfitting and consequent extreme sensitivity to noise and variations. We don't want to fit the noise, but the overall problem in order to truly satisfy our criteria. Notice that what we consider noise will depend on goals and success criteria. In attempting system identification, seeking a model for a component black box subsystem that correctly predicts and even previously unobserved behavior, solving the inverse problem of F inverse to parameterize component subsystem parameters x chi of that component, C, that's the bracket is C for this particular component. The inversion of the SVD assigns significant power to the smallest eigenvalues of the forward problem, and those are the ones that are most affected by variations in noise. Consequently, robustness principles of the neural coding involved within the subsystems and the constraining functions that determine that neural code are of paramount importance. This means that while one can attempt to solve component-wise black box problems independently, one should include a constraining process at the system level, such that the system identification is directed towards satisfying overall emulation success criteria CG, even if at the cost of component fidelity. In practice, this is probably always justified by limited actual in situ dynamic data collection at the component level, meaning that black box solutions at that level necessarily involve assumptions and estimates with broad error bars, as well as being justified by the combined effects of neural coding constraints and system noise. Another way of looking at this is to say that iterative studies aimed at eventually achieving correct brain emulation so that the principles of Axiom 1 are adhered to involve successfully better approximations or understanding of your specific goals, G, success criteria, CG, associated focus level, LG, and consequently, the actual forward problem that is to be solved. Doing this clarifies which observations, data, are in fact relevant and necessary, and which ones distract from discerning a useful model in the context of G. That brings us to scale separation. And sorry, I'm losing my voice, unfortunately. In complex systems, scale separation means that different parts of the system operate at different scales, either in time, space, magnitude, or all three. This allows separate analysis of phenomena at each scale without needing to consider all the details of smaller scales when studying larger ones. For instance, in computer science, you can study program execution without having to consider the properties of the analog transistors used in integrated circuits. Scale separation is typically the result of a combination of system structure and other constraints. The result of scale separation are useful protocols for signals and systems that are less affected by variability due to noise or other disturbances, protocols that are more reliable and robust. Evolutionary selection pressures have resulted in systems that are sufficiently reliable for the duration of the characteristic survival span of a specific organism, and an animal's survival depends strongly on reliable processing of sensory input, reliable recognition of threats and opportunities, reliable learning, and on taking reliably beneficial response actions. Although complex and chaotic analog systems will often exhibit fractal patterns and some degree of self-organization, in general, their trajectories can be highly divergent, highly sensitive to initial conditions and noise, and prone to unpredictable random walks in terms of states and high-level behaviors. It's worth noting that a small degree of randomness can provide a benefit to learning algorithms in that the disturbances can be a means of escaping local optima while searching for better solutions. Nonetheless, wild random walks at the system level are unreliable and dangerous. As will be explained later, the existence of scale separation is crucial both for the forward problem, the brain, so that it guarantees robustness, as well as for system identification and emulation, which is then theoretically possible and one hopes practically feasible. Unsurprisingly, we find in the brain that there's a large degree of systematic architecture, repeated occurrence of templated circuitry, and an application of robust signal protocols that are enforced by constant constraint mechanisms within each protocol. While the brain is a result of evolutionary pressures, engineers design robust and reliable computing must also impose scale separation when working with naturally analog components and have to operate in a variable environment. For instance, they do this by specifying voltage thresholds, including parity bits, imposing clock cycles, and much more. So for this, I'm putting down Axiom 2. Robustness requirements demand scale separation, and this must be true for brains. Just as scale separation is vital for the evolved system to operate reliably, it's also essential for its successful emulation. Scale separation allows us to replace parts of the substrate with mechanisms implemented in a different substrate. To do so, you have to find where scale separation occurs at relevant levels in this pyramid of mechanisms upon mechanisms found in nature. Above the relevant levels of scale separation, that which you wish to emulate is then substrate independent. Identifying hierarchical points of scale separation in the context of goal-directed success criteria within various functional subsystems of the brain can allow you to clearly express the problem that's to be solved and to selectively explore those methods of data analysis and interpretation that may find its solution. As will be outlined further, it's important that the chosen criteria, CG, are such that meeting those criteria can be confirmed by applying validation metrics, MD, that rely on measurable data, D. The data needed in this case, in some cases, can be identical to the observations at the focus level, so then D is Y chi, but that's not guaranteed, especially when success criteria belong to what I might call the emergent category. In those cases, the relationship with measurable data should at least be strongly correlational, and a set of multiple metrics may be required to together validate a criterion with confidence. We can summarize the process of mapping success criteria to data requirements as follows. You have a proposed description or representation. There's a corresponding identified structure. You have a proposed process for model functionalization. There needs to be data to make both structure and functionalization possible. You find types of measurable data that are well correlated with both. and these are then useful and necessary data to collect. Consequently, we could say exploring the right data acquisition depends critically on success criteria. The process summarized in the previous slides is non-trivial and definitely requires extensive experimentation. To reach a sufficient understanding, there's a meta problem that needs to be solved. We can express this as solving the problem ym is fm of xm. I know this is a little bit silly to put it in equation form, but hey, it's to make it correspond with things before, which is to be achieved by means of some overall scientific research protocol that's applied iteratively, thereby closing an improvement loop that eventually results in an accurate problem statement. In this expression of the meta problem, I'm calling the ym solved systems described using an appropriate hierarchy of instances of scale separation, and xm the useful and obtainable data sets as determined by requirements of that scale separation again, and fm are then groups of appropriate and successful validatable analysis methods. So above, we addressed reasons why total depth emulation is almost certainly neither feasible nor desirable as an overall approach to brain emulation. Nevertheless, approximating total depth emulation in very small organisms may be a necessary investigative process by which you can achieve the level of understanding of neurobiological processes and their interactions that are needed. Referring here to the Haspel et al. Paper that Conrad knows very well. One may be able to satisfy the requirements listed in Axiom 1 while anchoring the biological ground truth of a system so that is so small that optimizations addressing what I'm calling the trees of its neurobiological components don't lose sight of the forest of success criteria. An organism like C. elegans has a limited number of behaviors after all, and cellular and molecular connectomics addressable with current technology. The caveat is that the study of C. elegans, while valuable grounding endeavor, can't necessarily guarantee developing methods that satisfy Axiom 1 for a different nervous system. And here's a quick reminder of that axiom. Especially when larger brains rely predominantly on different neural coding schemes. This applies to mammalian brains, but even to the 140 neurons in Drosophila brain. Strategies that encapsulate neural coding in C. elegans fail to align that well with the relevant separation of scales, where the reliability of larger brains may depend on patterns of spiking activity in populations of neural cell types. Most cells in C. elegans don't spike. Subthreshold input does play an important role in modulating and synchronizing role in brain regions of larger animals, but their neurons typically require input in the form of bursts or synchronized input from multiple sources to propagate information by firing. Activation in neural populations is typically controlled and kept in check by recurrent inhibition, again caused by synchronous input from entire populations of interneurons. In many cases, recurrent excitation establishes attractor properties and cued recall of patterns of activation, and so forth. So on a roadmap with studies in C. elegans and other small organisms with similar neural coding strategies, one would expect that a subsequent progression to even more complex systems, taking along mechanistic insights gained, but reevaluating analysis, system identification, and emulation methods in the context of new success criteria and associated validation metrics is necessary. Another way to conduct investigations anchored directly in the ground truth of neurobiology is to study and attempt local, sort of something like total depth emulation of small subsystems that consist of in situ or ex vivo tissue samples from particularly well-studied areas of the brain. In this case, the best candidates are subsystems for which a full range of behaviors can be recorded relatively easily and that are largely insulated from the contextual complexity of the rest of the brain, so that their operation isn't significantly altered in the absence of the normal flow of those context-dependent interactions. Predominantly, that leads to candidates at the very periphery of the central nervous system. Examples are studies of sensory processing in the retina, cochlea, and perhaps motor control studies in brainstem, basal ganglia, or cerebellum, although motor control in all of those areas already involves significant recurrent interaction with neural protocols in other brain regions and may therefore be an insufficiently constrained target. One way to address the problem that validation metrics associated with success criteria require ground truth information with sufficient understanding to identify the hierarchies of scale separation to satisfy axioms 1 and 2 and that such of that understanding may be missing or incomplete in most cases, is to provide a controlled space for methods development through synthetic data. Even when methods development includes validation by application to biological sample data, synthetic data is inevitably used in the early steps of modeling and algorithmic methods exploration. Doing that is already common practice throughout computational neuroscience and machine learning. In place of one-off algorithm testing with synthetic data, you can design systematic protocols aimed at solving the meta problem by subjecting analysis and interpretation methods to a series of successively more sophisticated tests, evaluating their performance in the context of system identification and for the discovery of hierarchies of scale separation at every stage. Many examples are then used to improve the odds that measured performance satisfies criteria CG. Improving the set of success criteria themselves becomes a part of the exploration loop, and derived insights and well-performing methods must then be tested in biological systems. There are, of course, risks. One of these I'm calling misdirected performance optimization. If a specified set of success criteria is insufficient, or if emergent criteria are not sufficiently tested by corresponding validation metrics, then offering a challenge and performance scores will lead competing methods or algorithms to optimize performance in a manner that could be misleading. The second risk, drawing conclusions based on what I'm calling alien ground truth. Our understanding of the world of communicating neurons is incomplete. We don't yet understand enough about information processing mechanisms at the molecular level, nor do we yet know how signals at other scales interact in every region or circuit of a brain. In other words, our understanding of relevant neural coding can't yet reflect reality. It's unavoidable that generated example data will be based on incomplete or incorrect assumptions to some extent. Another way to explore scale separation and system identification in neural systems by means of ground truth substitutions is by using in vitro cell cultures or brain organoids. These biological but synthetic example systems may be allowed to develop structures without particular functional goals, or their development may be directed in a variety of manners, such as through the use of microfluidic devices or electrode meshes. A population of neurons in cell culture or organoid can be small enough and grown in a manner where the morphology can be imaged completely and dynamic activity of every neuron recorded. Stimulation at the resolution of single cells is also feasible for every neuron. The level of control is a bit lower, and the turnaround time for batches of experimental systems a bit slower and potentially more costly than in the case of in silico synthetic systems. But a greater degree of the biological ground truth is implicitly given, even if composition, arrangement, and context are nonetheless different than in situ or in vivo. So the critique for synthetic risk 2 applies because example systems will exhibit behavior in ways determined by the contributors that were selected for inclusion, such as cell types included, the number of cells and their density, duration of the in vitro development phase, the resulting structural arrangement of cell populations, modulatory input, and neurochemistry. A final entry point for further study of the meta problem is by means of a top-down strategy that begins by proposing and developing a battery of well-identified and categorized success criteria, by then focusing on constructing validation metrics, MD, where the data D can be feasibly recorded, and keeping in mind that emergent criteria may require multiple metrics as explained above. The resulting requirements can suggest directions for the development of system identification methods. The box on the right shows one result from processed surveys among stakeholders with 12 criteria in 4 categories. While this approach is rather different than the previous one, what it does is to emphasize a focus on system-level criteria, prioritizing that over ideal fitting of component black boxes. At the same time, it makes clear that there are many kinds of success criteria that need to be satisfied together. Okay, in conclusion, in this presentation, what I tried to do is I tried to give a cautious account of challenges and methodologies involved in system identification within the context of brain emulation. I tried to point out the gray box semi-physical modeling steps and attention to constraints and context. I emphasized the critical role of scale separation in both the brain and its emulation, the importance of paying attention to hierarchical points of scale separation and corresponding abstraction, clear goal definition corresponding appropriate levels of scale separation should guide and be the first things to be explicit about in functionalization or emulation attempts. I also pointed out the iterative nature of the meta problem. And with that, I thank you for your attention. And that's it. Thanks.
So Margot Hanley asks, I don't have a background in this space. I work neuroscience in the context of AI and neurotech. I'm wondering if there's a way to think about the problems of brain emulation presented here in relation to contemplating, complementing the recording slash predicting of neural activity with brain foundation models.
Before I can properly answer that, I have to understand a little bit more about what is meant by the recording of activity with foundation models. Or was it not stated as with foundation models?
I can jump in here.
Yeah, sure, go ahead.
Hey, thank you so much. These were fascinating presentations, and it's not my space. So, but I think I'm working in a tangential field in AI neuroscience and commercial neurotech. And I work a lot on the question of like ethics of consumer neurotechnology. And right now I'm thinking a lot about foundation models, and particular creation of foundation models trained on brain data, and the ability to use these models to do all the things that these foundation models can do, like diagnose certain things, and then also kind of create neural data that technically could represent that individual person. And so I cite in kind of these questions I have here, like first person brain foundation models that companies or researchers are trying to create, where you have someone where like a rig that's collecting 4 or 5 different data types to create effectively a way to make a rich portrait of that individual, and with some 60,000 hours be able to use that foundation model to both record and technically predict a lot about that individual. And I'm thinking about this set of techniques used in a really different setting, right? I know it's not neurobiology, but with the attempt to understand the brain and kind of these mechanisms in a much deeper way, and kind of how that relates to the work of brain emulation you're talking about today. And again, maybe it's too far afield, but it just seems like a really different technique to try to understand potentially similar mechanisms, or maybe not. And I suppose that's the question.
Mm-hmm. Yeah, sure. So there is something that I didn't address in this talk that I should probably get into here, which is, so if you're using a functional approach like that, rather than one that's based on connectomics or some other method of really digging into the brain, then you're basically declaring the entire brain or the entire person a black box. So you're not using really small black boxes. You're using, if it's MRI, then your black boxes are whatever the resolution of that MRI is. If you're using EEG, then it's another resolution yet again, depending on the number of electrodes that you have, and so forth. So the problem then is that you have a really big black box with a whole bunch of complexity inside of it. And so then the question is, can you state this as a problem that is not ill-posed? Is there a way that you could end up with a solution where you don't have many different models that could explain the same data? Now, for some utilities, for some applications, that doesn't matter. And I think that's kind of what I was trying to get at in this talk, is that there's always this connection down from what are your goals, what are your success criteria, and therefore, how should I approach the problem? So if what you're trying to do is, for example, build an online agent that behaves like a certain person, that's an entirely different problem than, say, a medical application where you're trying to build a neuroprosthetic for a piece of or an entire brain, and the internal states should also reflect, say, for example, subjective experiences of some kind. So I don't know if that answers what you're saying sufficiently, but I think that's an important thing to keep in mind as well. So it's really, what I'm trying to get at is this, trying to state your goals clearly. And one thing that popped out at me, that came out, that I was noticing in listening to the previous talks, was also that really coming into this and just saying, functionalization or brain emulation, or using a term like that, we get into that and then we are discussing some approaches. And in the case of Phil and Jan, they were in a very similar subdomain. But you could also come into this with different goals. And really the goals that you have determine to a large degree what is satisfying, what is a good outcome. and that then has a huge effect on everything else.
That's really helpful for me, Randall. Thank you. Maybe I'll follow up separately, but thanks that does answer a large part of the question. So thank you.
Thank you.
So Margot, I know you followed up with what isn't technically a question. Was that covered in his response?
Yeah, that was kind of just like that first person foundation model paper that I've been thinking about that seems to be pursuing it, that I supplementary information, if you will.
Cool, thank you. We have a question from Alexander McLean. They ask, I didn't quite understand this. Are you saying that separation of scales break down for C. elegans organisms? Is there evidence that separation of scales hold and can be used to our advantage in other organisms such as human?
No, I didn't mean that. No matter what organism you're looking at, you can always say there is some separation of scales that you care about. I mean, in a completely analog system, maybe whatever, some system where it just doesn't apply. Let's not talk about those. But if you have C. elegans, then your success criteria are to, for instance, be able to mimic its behaviors. And then the level at which that takes place may rely on the activity of very specific neurons doing their thing, because C. elegans has so few neurons that in there, every neuron is almost like its own brain region doing a specific task. And then they have to interoperate in a fairly analog way. So you can still break that down. You can still say, okay, there, the level of the scale at which I have to look at this might be a very careful modeling of that cell and its metabolics and various other things. You might have to do that in this case. Now, at another level, let's say you're looking at Drosophila or you're looking at the mouse. Let's take the mouse. You may have an area of that brain, say the temporal lobes of the mouse, where there is population activity and there are all kinds of recurrent inhibitions going on, so that a bunch of what would otherwise matter at the level of an individual cell is cut off, drowned out, inhibited, not relevant for the function of that little piece of subcircuit, or not as relevant up to some threshold. So you can have a different type of scale separation depending on the animal that you're looking at or the problem that you're trying to figure out. So the goal that you have for this. So even in, say, mouse, even though you're looking at populations in many cases, it could be that you care about pathologies. So if your goal is something like Alzheimer's research and you're using a mouse model for that, then the scale that matters to you is the one where those pathologies have an impact. So then you may have to model metabolic aspects, for instance, of cells that might be affected by whatever that process is that's affecting it in Alzheimer's or Parkinson's or whatever. So it's the goals and, of course, the system that are different. And so that all impacts what kind of separations of scales at different hierarchies you might run into.
Someone asks, I realize this may be tricky to do, but could you zoom into how the inverse problem applies between C. elegans, fruit fly, and mouse brain? Are you proposing some sort of, quote, universal language to apply for each emulation?
Well, I'm proposing an approach for study. The inverse problem and its detailing, I did this mostly as something to fall back upon so that you can say, okay, we have a reason why we care concretely about these different things, why we care about the criteria, why we care about the scale separation. So it's kind of a buildup of a reasoning of why should we care about these things in a certain way. But what I'm then ultimately at the end of the presentation, I got into different ways that you could address trying to take apart the problem, what platforms you could use, for example, what study platforms you could use. And that's where I'm making a proposal. So I didn't point that out directly because there's, the introduction to the Brain Emulation Challenge coming up later. But what we're trying to propose is that even though you can't have biological ground truth when you're generating synthetic data, that there is a place, a useful place for having a platform that can very systematically explore a space, a subspace of the possible difficulties you can encounter, say the number of cells, the density of cells, whether gap junctions are included or not included. So the complexity of the system you're studying, the influences that might matter based on your problem. So based on the specification of your success criteria. So for example, do you care about things that matter in pathology, or do you only care about function? Is that what you're trying to look at? Is it something even bigger than that? Or are you just caring about, say, subjective experience, if you can put that in a model? That it's useful to have systems where you can explore the methods that you would apply. So methods like the ones that Jan and Philip were using, and then alternatives to those, additional approaches, ways of analyzing and interpreting the data that we've collected or data that you could collect in future, and that you can kind of go through them, sift through them and explore them in advance to some degree, at least to know, well, this method is going to be very strong here. This other method is going to have certain weaknesses, and then apply that in biology and go on from there. And then, that's why I mentioned there were some risks involved, obviously, but as an iterative process, we're proposing that is an entry point. So I'm proposing that, yes, you can tackle C. elegans, Drosophila, mouse, and they have different levels of scale separation. The goals that you might be addressing are different, but there can be platforms, such as the ones I listed there, where it's useful to then carry out studies that help you develop methods so that you can improve how much you can get out of the data that's been collected or will be collected, and speed up the process a bit. I'm very interested in any way to accelerate progress, basically. So any methods or any protocols that help with that are of interest.
So Adam, I see your point about the ground up approach, but the response is quite long, so I'm not sure I can do it justice just by reading it out loud. Is this something you'd like to talk to Randal about directly?
Okay, thanks. Yeah, so I was wondering really just to approach it from the ground up, really to come up first with a single cell, whole cell simulation model and build on that, right? Just to really start with, let's say, with MD simulations, determine how the individual proteins react, and build some, let's say, PD models of cells that can be parameterized with 3D structure and their epigenomic state, right? And this is something that can have huge applications in, let's say, drug research. So of course it's a big task, but maybe if enough companies would work on that, we could get to something. And so then you have this basic cell model that we can parameterize for the specific neuronal types or other cells in the brain, and then gradually go up and validate against actual biology, for instance, with organoids. So I was thinking, do you think this approach is feasible, or what is the?
Yeah, actually, I'm very interested in what you're saying right now. I'll say again, to try to put everything in the right context, that my interest is mainly in understanding the brains that exist, is in understanding how to take them apart, replicate them, build them, basically. So that's why I've came at it from that approach, and then worked my way towards protocols or platforms that could be useful. But what you're saying is directly in that ballpark. It's in that area, in this domain of building a synthetic system, in your case, all the way from the ground up, and then using that synthetic system for some purpose. Now, that synthetic system could be used to then test ways to explore whether or not a particular method can pull out what you need for a recreation, a functionalization, and emulation, which is my particular interest. And I'm sure there are other applications of it too. But yeah, so that kind of synthetic building from the ground up of a neuron, a system, is pretty much what we're trying to do, and what will be discussed more this afternoon. Although we don't go quite as deep as what you were saying, not yet. There's nothing wrong with going there, and we're totally interested in also diving down to that level. It's just that we had to start somewhere for prototype stuff. So, but yeah, that's exactly what I was talking about. Yeah.
Right. Thank you. Yeah, and if anybody knew about some like commercial like job with reasonable salary where one can work on like wholesale simulation, I would be very interested in that.
Okay, cool. I think what you should do is you should join our Discord forum where we're doing, that we're offering to everyone for post workshop discussion, because then, yeah, something like that might come up.
Thank you.
So Randall, in your slide about scale separation, you mentioned wild random walks and how that might be unreliable or dangerous. Could you maybe elaborate on that?
Yeah, sure. If you have a highly complex nonlinear system that isn't constrained in any way, it will probably have some features that pop out because of how that complex system happens to be developed. That's what I was mentioning. They could have like this fractal output in a sense. But at the same time, whatever that is may not be sufficiently constrained and was not tuned in a sense to the problem of survival. So for example, if you have one brain region where there are a bunch of neurons just operating as analog components that are unconstrained, if there are very many of them, there's a high chance that they may go off into some operating domain where they're oscillating through something like with some rhythm, or they're ending up in certain attractors. Now, attractors are good, but they can be good, but these might not be the attractors you were looking for. And similarly, the other system that's trying to communicate with it, it's supposed to have some kind of a function, in this case, in the human brain, may also be going through all that kind of stuff. So if they're together trying to identify whether that thing in the bush over there is a tiger or your grandmother, you don't want that to be a misidentification. So it's very important to have systems that communicate with one another reliably in the brain, and then even smaller, like subsystems, circuits, etc. And generally, if you want to get something very reliable and robust, the more you add certain constraints that make sure that these systems stay within their operating specs, the better. So that's pretty much what I was getting at. This is a sort of general way of talking about it.
Yeah, thank you.